মনে হচ্ছে এসে পড়েছে। এবার ঠিক আছে না? মিউট করে দিতে হবে। Okay, welcome back. Our last day on with HPC. আর শীতকালে তো একটু ঠান্ডা লাগবে আর কি করা যাবে। এটা নিয়ে হ্যাঁ। So we had to change plan a little bit. The reason is like today will be my last day. I wanted to stay until tomorrow, but wherever you are going in Sundarban, there is no way to come back, return from there. There is no travel arrangement from that deep. So I'm going to get off the boat at 9:30 p.m. today. So that, yes. So what we'll do is like, we'll cover some lectures in the boat. We'll have a discussion about everybody's project and project interest and how we can, I can give critical feedback and so on. So we'll do that. And then Sabrina will be, there'll be a little bit of MD analysis that Rahat and Shishi will cover tomorrow. And then Sabrina will cover the genomics part. So I think 4, 6, 8, 9. We are missing 4, 5 people. Okay. So the first thing that before we start today, I want to make sure that if you have generated the trajectory files that Rahat put the instruction on. So Rahat is going to share his screen, and if you log into the OSC on demand, so why don't you log into the OSC on demand?
So all of you have done the instruction from MD task file. The MD task file I have posted it in the Slack. All of you? Is there anyone who didn't?
Yeah, so we can go, yeah, let's go slowly. So why don't we log into the Ohio Supercomputing on demand, and we'll check who has,
I have not, but that's all right. I'll catch up later. This is on YouTube.
Oh, hey, is that Brad?
Yes.
Okay, yeah, don't worry about that, Brad. That was from the first day. And yeah, so it's for here because we are, next 2 days, we'll be doing it without internet. That's why I'm making sure everybody did that. Yeah, don't worry, Brad.
All right, thanks.
Okay. So some people may not yet follow the instruction from MD task file. Okay, let's see.
So why don't we do that? Let's go. We'll check here step by step. Rahat will show where you look for the file.
Yeah.
Whoever doesn't have the file, Rahat and Shishi are going to help you to submit those commands now, and those file will be generated in an hour. So you'll be ready to go. Okay.
Yeah.
So have you logged into the Ohio Super on demand?
ondemand.osc.edu.
Okay, good. So if you go there to the files.
Yeah. And then go to the home directory. You see your user directory here. User PGS 029 and then username. Okay, and then go to the CompBio directory. And then charm GUI 76 or whatever your number is. Okay, and then go to name directory. Okay, you see a folder named step 4. Can everyone see that?
Yeah, they should have a step 4 folder we
Yeah.
generated. Let's go.
Okay.
Let's go inside the step 4.
Yeah.
Okay, so let's wait here. So what you are doing is you are going to the on-demand OSC, and then you're going to the file. Under the file, you look for your home directory, then CompBio, then charm GUI, then NAMD, and step 4. That's the folder we will be, right? And if you have the files inside that, this file, all these files, that means you have finished your run. If you don't have these files, that means that those MD simulations either you did not run or they did not generate the
Output file. Yeah.
So what we are going to do, if you have those files, you are going to, I'll show you which 2 files to download to your laptop. You'll download to your laptop. If you don't have those files, then Shishir and Rahat are going to go help you to run those so that we have those files before we leave today. Okay. So,
okay.
Can you go to the DCD? Yeah, just stop at the DCD file.
Yeah.
Okay. So this is the file that's, if you remember, I told you that is the most important file, .dcd file, and look at the size. So you should have a big size. I mean, not big enough, but bigger size. Okay. If that size is small, that means like something happened, the output didn't come through. So let's say how many of you don't have a DCD file in step 4? How many? 1. Oh, good. 2. Okay. That's less than 10%, 90% success rate. Great. Okay. So, so those Shishir and Nath, you can help them. Okay. So that they can start that. And then now, if you click on here, in that button, you have the option to download it. You can download. So please go ahead and download that file in any of the directory that you want. Okay. And then the other file that I want you to download. Okay. So let's start that. The other file the PSF file. The other file that I want you to download is one step up. You have to go back to the previous directory, right? At the NAMD directory. So the way to go back to the NAMD directory, you can click the NAMD directory. And then if you move up slowly, there should be a file called .psf, this one. Okay. So that's the other file I want you to download. Okay. Yeah, PSF. So does anybody remember what is a PSF file? Okay, who said that? Good. Protein structure file. What does it contain? Okay, you already got 80% number. Now for next 20%, what does it contain? Sequence. Okay, so what protein structure file, just to reiterate, what it does is like, it takes the atoms in protein and it says like this atom is like this kind of atom in force field parameter. So it just connects those two. And I hope we get a time to look into it. Okay. So those are the 2 files you need to download. In the NAMD directories, the .psf, step 3 input.psf. You can, I mean, if you want, you can download these 2. Let's say, let's download for safety. also the PDB file. Okay, so those are the 3 files, and we will analyze those files on the board. And if you have those files, then we can do it. Okay. So should we take a few minutes for you to download? Okay, let's wait a few minutes for you to download. Input.psf. রিহাদ আর ওকে দেখো রান করা নাই ওদের দিকে ওনার আছে ঠিক আছে তোমার এটা তোমার স্টেপ ফর ডিরেক্টরির ভেতরে এটা অন্য কোথায় আমরা আগের দিন যেটা রান করছি স্টেপ ফরে দেখো তো স্টেপ ফর ডিরেক্টরি থেকে যাও তো
So the DCD file should be in the step 4 directory, not outside of step 4 directory.
Okay. So you may have a DCD file outside the step 4 directory.
Yeah,
that one was when we generated it in our class, right? And that's the smaller directory. But the one that we want to analyze is where he put an instruction in the Slack. It's okay. We can go through that instruction. It's a very short instruction. So let me then ask, how many of you have a DCD file inside the step 4 directory? How many of you have a DCD file inside the step 4 directory? Okay, 1, 2, 3. So 4, 5. So maybe we should go through that step. So let's open the Slack. I'll open. Thank you. Okay, so let's, why don't we do it all together?
Okay. Let me.
So let's go to our Ohio On Demand and let's open a terminal.
Oh, okay. Yes, please. So you have to go to the oac.ondemand.a2 website and then ON's shell access. Okay.
আমি এখান থেকে অন করতেছি, তুমি এটা শেয়ার করো। আমি
Slack.
পিডিএফ ফাইলটা এখান থেকে খুলতেছি।
Okay.
খুলতে পারবো। ইয়েতে দিছিলাম না
Slack a course.
Slack. Okay, so the instruction that we are going to follow was on the Slack called mdtask.pdf. And since Rahat is sharing his screen, I think you can either open that on your laptop and I will, তুমি এখানে চলে আসো, এটা দেখতে পারবা। হ্যাঁ, তুমি এটা দেখে ওটা ফলো করতে পারবে।
So I'll be just following these steps from MDTask file. So first, open a shell access from ondemand.osc.edu. Yeah, open clusters and then ON shell access. Everyone can open ON shell access? Great. Okay. So if you type ls, then you should see something like that. CompBio on demand and some other files maybe. So can you see the CompBio directory there? Everyone? And then cd into the CompBio directory. Cd, that means change directory to CompBio. cd CompBio. Let me clear my screen once again. Yeah. CompBio. And then cd charm tab, this one. And then NAMD. So I'll change my directory to NAMD folder in the charm GUI generated from the charm GUI website. So if you are in NAMD directory, you should see something like this. You should see a folder like step 4. Are you seeing that? Everyone? If you are not seeing the step 4 file, step 4 folder, raise your hand. No one? Okay. And then open step 4 underscore equilibration dot in using nano. Yeah. Okay, so if you open the step 4 underscore equilibration.in file, so in the set output line, there is 6th line, you will see that there is only step 4 underscore equilibration, but I need you to change that like mine. So step 4 equilibration will be step 4 underscore slash step 4 underscore equilibration. Yeah. Dollar home. Yeah. Variable. Right.
So in this one, what is saying set variable name is output name. So set a variable and its value is this one. The reason is next time you want to use this long thing and you go down this you don't have to go. Like this dollar sign output name is something. All the files that you will display, they will use this value. That's why you can see the step 4 to be on, and you can see the 4 on that we have set up here. So that's the so that's why you did this step, but it's good that this is how you decide where you are going to put your file. If you want a different directory, you come and change here. And that's what we are doing.
So everyone done with the 6th line? Okay. If you're done with the editing, press Ctrl X. And then, yes, I haven't changed anything. That's why it's just stop and exit it.
So yeah, you have to sit Ctrl X,
And
Y,
then Y,
and then
and
enter.
enter. Okay. So-
Wait, I think it's still doing something there.
Everyone done with the editing? So everyone done with the editing? How many of you still can't edit the equilibration input file? Raise your hand. If you're not done with the editing. Okay. So next you have to go to the step 4 directory. So CD, that is change directory to step 4. So if you type PWD, you will see that at the end there is step 4. So you are in the step 4 directory. Okay. Now I want you to open step 4.sub file by typing nano step 4.sub. Okay, before that, everyone should be able to see the step 4.sub file. Is everyone seeing the step 4.sub file? Yeah. If you type ls, then you'll see step 4.sub. Everyone seeing this file? Anyone who is not seeing, raise your hand. Okay, so everyone is seeing. Now open the step 4.sub using nano step 4.sub. Internet is a little slower. Oh. And then press enter. Okay. You should see something like that. I want you to change the time parameter here. You should see something 12 minutes, I guess. Yeah. Yeah, you should change your parameter. I think it will be 12 into 40. Oh, so Is everyone done with the editing? How many of you haven't write 40 instead of 12? If you are done with the editing, then press Ctrl X. Yeah. And then Y. And then enter. Okay. So after that, you have to submit it in the Slurm job scheduler by typing sbatch step 4 dot sub. So type sbatch space step 4 dot sub and then press enter.
So if you have submitted, then we start the next part. And after that feedback, we can come back and download. Okay, so if everybody has submitted, we would like to move to the next part because we'll have another quick hands-on that we'll submit something and we'll wait for it to be run, okay? So everybody happy with the MD part? Okay, so we are now, the train is going backward. We run the dynamics, now we are going to do the modeling. Okay, so even for the modeling, we'll do the hands-on first and then we'll explain the theory. Okay, so it's all going backwards. And so hopefully you will have. So do you have Chimera? Okay, well, let me step back. So we are going to predict structure of a protein, okay, using AlphaFold. And the prediction of AlphaFold, to use AlphaFold, you have 3 different options. One is to run at the OSC on demand. That is a little bit more involving because you need to type all the commands. So we are not going to do that. However, if you are feeling comfortable with your command line, you can go back to the OSC and you can run. The second and cheap option is if you have a Gmail account, Google account, you can go to what is called Google Colab. And Google Colab is a collaboratory laboratory space by Google where you can run calculation on Google computer. Okay. And they allow you, I believe, to run for 30 minutes or something at a time. And for smaller protein, that should be enough to predict. Okay. And another even more easier option, even easier option is to run through Chimera X. So if we predict through Chimera X, what Chimera X does is it runs the Google Colab for you and then fetch the data. So it's all in a one nice package. Okay, so for today, if you have Chimera X, we are going to use Chimera X. If you do not have Chimera X, we'll use Google Colab, and I will show you both. Okay, and they are very similar. Okay. So if you have Chimera X, then you can open Chimera X. না থাকলে আমরা Google Colab. So if you have Chimera X, we'll use Chimera X. If you don't have Chimera X, that's okay. You download it. We'll use Google Colab for now. I'll show both. Okay. So if you search, open your browser and search Google Colab. Oh, it's too small. Let me open. So, I mean, maybe all of you can use Google Colab. What I can just demonstrate in the Chimera X what additional things it makes. You know, if that's easier for you, all of you can use Google Colab. So if you search Google Colab, the link will come, and then let's see if I can hide this back. Okay, I mean, for me, because I was using on it, I have a different, you know, I was using some of the Google Colab. So what Google Colab does is like, it allows you to write program in Python. And then anytime you see IPYNB, that means like Python notebook, and I might be for interactive. I don't remember what is the I for. But the underlying language is Python, and it's a Python notebook. And that's what they use. And let's cancel this. And you need to log in here. I do not know if you log in, if you're already logged into Google. If not, you need to log in there. Okay, so once you log into Google Colab, and if you haven't, you know, used the AlphaFold notebook before, it's, you know, we have to search for it, right? And the way that I search is I go open another Google, another Google search and look for and the first one does come as AlphaFold. So you have to log in. That's why I opened the Google Colab first. But now if you click on the AlphaFold Colab, and that's okay. And, you know, there is a 30 years of work went behind it to develop this thing. And, you know, it's an awesome thing. It's changed the whole field. It looks very unassuming, but trust me, this is one of the biggest discovery, at least for the decades, okay? Biggest invention. So are you on AlphaFold Colab? Okay, good. So we are going to predict a protein. Actually, we're going to predict a protein complex in the today, okay? And you can go ahead, use your own protein later. The reason is, if you give too big of a protein, you have only 30 minutes of runtime, you may not finish it on Google Colab. Okay, so that's why it's we are going to use a small, smaller protein so that we can finish the run. So, but before we predict, I want to tell you, like, Google has already predicted, I don't know, a huge number, millions of structures already put in their database. So it's a big possibility you don't have to predict it. Okay, so but we'll still go through the prediction, then we'll go to the Google data AlphaFold database. So all you need to do is put your sequence here. That's all you need to do. And if you want so if you go to the step 3, you need to put your sequence. And if you want multiple proteins, so you put one sequence on sequence 1, another sequence on sequence 2, and so on. Okay, like if you want them, if you think like those proteins are going to interact. Okay, so I'm going to fetch a protein sequence, and I'll show you how I do it. But before that, I have a question. If you want to know about a protein, know all the data, how do you know? It's not a question, it's a feedback I want. Let's say your advisor, your professor says like, we have this new protein. What information is out there? How do you find it? Okay, one. So what do you do? Here.
Well, if we have the sequence of the protein, then we can compare it to the protein database so that we can find if any similar type of protein had been annotated or its function had been worked on.
Protein, PDB, protein data bank. Okay, and anybody else use any other thing?
It's going to depend on what you have. If you have a sequence, that's going to be different than if you have a structure or maybe some other data about what it is.
Well, just wait for my answer, Brad. I think you might have everything together. I'll just show you. Yes.
There is also another database called UniProt.
Okay, that's the answer I was going to say, was looking for. So if you have a protein and you want the information, then you should go to UniProt. Because what UniProt does is it combines all the information under the same website. And we should have a workshop just on UniProt because it's such a rich, and we do offer workshop just on UniProt. So let's say I want to predict ferritin, that's a small protein, and ferritin has two chains, and ferritin is becoming very famous because ferritin creates a nano cage and is using as a vaccine delivery vector. So I'm going to go to UniProt, and we'll do a little bit of UniProt. So if you type uniprot.org, what UniProt does is it has anybody can submit their sequence if they find a protein and their information to the UniProt. Also, UniProt regularly search what new information about a protein came up. And then once those protein information, like sequence information, goes to their database, there are two aspects. One is there are real human scientists who go through those proteins. It's called human curation. And they say like, okay, this protein, this function, I trust it. And they curate. And there is another part called automated curation, where some algorithms, so those reliability will be slightly less. But there are people always working, and anytime you come up with a new protein, you're encouraged to submit it there. And if you go, they have, they really put together everything nicely, and I don't want to go there, but I'm just sure. Let's say I will try type ferritin. So if you see ferritin, ferritin from all different organisms came. I'm interested in the human, but the first one is human organism, Homo sapiens, and that's the light chain of ferritin, and then the heavy chain is the fourth one. Okay, so let's first go to the light chain one, and we will go through just okay. And again, I'm telling you, this is like a graduate student can all imagine what kind of information they collect during their PhD. It's all there. Okay, so on the left-hand side, what you'll see different aspects of the protein information, okay? Names and taxonomy, that's the first you are looking at. Subcellular location is very important. Even if in cell environment, if two proteins are not in a same compartment, even though they may interact, they're not going to interact. They have to be in the same localization. Okay, otherwise they are not going to interact with each other. That's why you need to have subcellular location. And if I click it, it's going to tell you where these proteins are located. And then disease and variants, if any disease is known for this protein, and also any SNP, any variant that's already in the literature. So you can go and look if there is already any variant of this protein, you can compare with that, okay? Post-translational modification. That means like after the protein has been synthesized, if there is a further chemical modification like phosphorylation and so on, you'll get at the post-translational, and these are all like nicely put, and the visualization is really nice. And then also you can go through it when you get time. The two parts that we are interested, one is structure and other is the sequence, right? So if I click on the structure, you will see this is what the structure you are looking at is actually full cage, full nanoparticle cage of ferritin, okay? So ferritin light chain and heavy chain, they're like two alpha helical structures, they come together. But if you go below, then you can look at the source, or even like go below, they collect all the structures that has been solved by experimental, okay? So you can get X-ray structures, and then I believe there must be some cryo-EM structure of this one. Okay, so there is a cryo-EM structure. And then since last year, what UniProt is doing is actually putting the predicted structure by AlphaFold as well. Okay, so even though we are going to predict it, we actually don't need to. UniProt has already uploaded the last line, you can see AlphaFold predicted. It's already uploaded it. And also AlphaFold has its own database. Okay. And then anything this is the structural side, anything that you want to do with the sequence side, like you want to go to NCBI and all this, you can actually go all of these when you click on the sequence, and they'll give you all the options of doing that. Okay. And you can blast, you can do other things. So UniProt, the first place you have a protein, you have a sequence, the first place you go is UniProt. Check it out, what others have done, and then you can decide like, okay, I need to do this. So we are going to copy. Yeah, Brad, if you have any questions, feel free to jump in. So if you click the download, we get the sequence for the light chain. And we're going to copy that and put it on the first. Okay, so the first one, I'm copying the light chain sequence to the sequence number 1. So what I did was in the UniProt, I went to, hold on. Oh, just paste in the sequence 1, the first sequence. Delete whatever was there before and just paste the sequence there. Yeah, let me go slowly one more time. So what I did was I clicked the sequence, then I clicked download. That just gives me just the fastest sequence. And I copied all of them, just the sequence part, right? Not the comment, not the all the comments. And then paste it here. Okay, we paste it. Okay, so now we are going to paste one more sequence. Okay, so the initial search that I did with ferritin on the UniProt website, I get all the ferritins. I picked up the first one, which is the light chain in the Homo sapiens. I'm going to now pick up for me, it's the fourth one. The identifier is P02794. So that's the heavy chain in human. And if I go to the sequence, I will do the same thing. So I'm going to the P07294 for the heavy chain of the ferritin. And I'm going to do, and then I want to copy the sequence of the heavy chain and put it on the sequence 2. Okay, so I think there might be there. I think because the copy and paste might create a problem. I'm not sure. So, but I do suggest there has been a space here between the line breaks. So just delete the spaces in the sequence line. There should be two space because of the line breaks. Right here. Yeah. So I'm deleting the space because AlphaFold may think that's an residue, unknown residue. So I'm going to delete there should be two line space where the line breaks are, and I'm deleting those. Okay. Let me know if you follow that. Oh, 3. Okay. So there should be 3 space. Just delete any lines, any space that you see. Yeah, I saw 2 though. Oh yeah, third one last. Okay, I see. Okay, so I think there are 3 spaces in the second one, and first one has 2 space. Okay, so yeah, let's delete those space. I'm not sure, it may not create a problem, but it may, so just delete those space. First one has 2 and the second one has 3. Okay, let me know when everybody's there. And then we'll run it and then we'll move on to the next part. No, if you only put 1, you are going to get the model of that one. So if you put only 1, it uses what is called AlphaFold monomer. That means it's going to create the model for 1. If you put 2 or 3, then it's going to create the model for 2 or 3. But you have to remember, AlphaFold is very good predicting monomers. If you put multiple sequence, it does, not necessarily you're going to get an answer all the time. Okay, because complexes are harder to predict. For ferritin, I know I'm going to get an answer. That's why I'm putting it. So most of the time you'll probably put 1 just because you need your monomer. But sometimes you can put a dimer too. Like if you know the same monomer interacts with each other, you can put the same sequence twice. Okay, so like sometime two protein molecules, the same molecules make a dimer, like protein molecules, or with another protein, they stick together. That's called a dimer. Now, if the dimer can be of two different kind of molecule or two same kind of molecule. Now, if you want to predict a dimer, it's called a homodimer. That means like two molecules are same. Then you put the sequence twice, same protein sequence twice. Then put it 3 times. That's why you have so many sequence options here. So if you know that your protein is a trimer, then put it 3 times. Like homotrimer, same protein 3 times, heterotrimer, yeah. Yeah, so in heterotrimer, let's say you protein A will go on the first, protein B goes second, protein C goes on C. Like whatever the complex you are trying to form, you just put the sequences. I'm showing a heterodimer here. Okay, is everybody with me now? Good. Okay, and the running part is very easy. Let's see, and I have to remember how to run it. Hold on. Okay, in the run time, if you go on the run time, if you just hit run all, that's all you need to do, run all. Yeah. And then it says like, this is not authored by Google, that's okay. It was developed by the community, run anyway. And if you now, well, for me, you'll see it's allocating, that means it's allocating the resources, then connecting, it's going to go through those. Okay, so it's connected. For me, it says connected. And you'll see each of the cells will be running one after another after another. Okay, so that's the end of the protein structure prediction. Okay. Yeah, it's simple, yes.
AlphaFold predicted structure have a long tail that we have seen earlier, and X-ray crystallographic structure that doesn't have.
Yeah.
So why it's happening and what the role of that long tail?
Okay. So the question is like, what's your name again? Shahadat. So Shahadat predicted an AlphaFold structure, and he saw a long tail, either an N-terminal or C-terminal, not structured. And X-ray crystallography did not have that. And we'll come to that. The thing is like, AlphaFold, this modeling has been going for 30 years, and we'll have a little bit of history of that. But lots of good algorithm, but none of that knew if their results are correct or not. They can predict, and they say like, one of the 10 will be good. But they didn't know there was no measurement of error. AlphaFold is the first one who has the measurement of error, in the sense that they can tell you how much confident I am in my prediction. And if you look at that, and we'll look at how to look at the confidence. If you look at the tail part, the confidence will be very low in this, because that means like that part of the prediction, AlphaFold doesn't know what to do. That's why it just put as a long tail. Okay, so that's what it is. And we'll see how to check those confidence intervals. Okay, so everybody's running their prediction on Google Cloud. Okay, then we can start the session that we are supposed to start at 8:30. Okay. Well, that's good. We all did all the hands-on work. So we'll start the lecture. We are supposed to start the day with a lecture. We can start now. হ্যাঁ ও হ্যাঁ ওটা ভালো হবে How many have you seen the movie, The Hitchhiker's Guide to the Galaxy? Okay, if you know, it's one of the funniest movie. You should go and watch it. It's a really nice comedy, British comedy, but they're really nice. One of the when Pradeep is looking for the pointer. One of the idea was really good that you can time travel. So just before the whole universe goes through the what is called heat death, like before that, all the entropy has maximized, so you cannot survive. You can watch the heat death in a restaurant, and you can eat as much as you want. The reason is because of the inflation, because you're traveling times, right? If you pay 1 penny right now, by that time it will be like $1 million. So you can eat as much, and you can come back and pay a penny. And that's like, it's really nice movie. You should see. Yeah, okay. Thanks. Okay, so let's see, is that the first one? Yeah. So what I'm going to do is like, I'm going to give a little bit of history at the same time. I'm going to touch the experiments that used in protein structure determination, because experiment is still, the gold standard of knowing a protein structure, and different experiments has a different philosophy. So we'll go through that, but we'll start from why do we model the structure? What is the underlying philosophy of modeling the structure, right? And it was a work by Christian Anifson from NIH. He got the Nobel Prize for this. The fundamental theory is, if I have a sequence, that sequence is going to predict a unique structure. So that's the underlying dogma. Okay, if I change the sequence, that will predict another, that will be a fold to another different structure. Because if the same sequence folds to 20 different structure, then you cannot do a modeling, right? You don't know which one it will go to. So that is the underlying dogma first, which is like a single sequence will create a single fold, unique fold. Having said that, that's not totally true. There are few proteins that creates two different stable structures. Like some of the disease, that Alzheimer's disease is one of the better known. There are similar disease where the protein in the cellular environment is alpha helical, which is a very stable structure, but it can go to a beta sheet structure, which is another very stable structure. But when it goes to the beta sheet structure, the cell doesn't know how to degrade it. So those plaques keeps growing and your brain function deteriorates. Okay, so there are few examples where a single sequence can exist on multiple different stable structure. But for most of the case, 99.99%, it's not. And that is the fundamental of dogma of why we do protein prediction. Okay. And then that's the dogma. And then there is then, your question? No. And there is then technological part, which is like sequencing has got super cheap. I think like sequence per base is like few cents now. If you send for a sequence, it takes like few cents per base to sequence, right? It got super cheap, super fast, and accurate. But the structure prediction is a very costly thing. So right now, if you go, and this was done, taken by last summer, I think if you go to GenBank database, you have 2.6 billion sequences because they are cheap. People all over the world are doing and uploading. There's a huge number of sequences. I think this number must be a mistake. Okay, so a good fraction, I think it's 200 million of them. 200 million of them is translated, that means like from sequence to made to a protein and deposited to UniProt. Okay, and as we went to the UniProt, that is a real treasure. So if you want to learn about your protein first, that's your first stop. And in protein data bank, you are looking at around 0.2 million. Okay, and that's after 50 years. Okay, after 50 years of work. So now, if you divide these by these, we are a factor of roughly 10,000. So for every 10,000 sequence, we have a one structure, right? And also, the cost of these is of the order of $100, maybe $1,000 to sequence a protein. The cost of an experiment of the order of $50,000, $100,000, right? So there is a huge resource gap and there is a huge number gap. And that's why we need to come up with the modeling strategies so that we can catch up with the sequence, because there's so many unknowns there. And the structure gives you the information, correct information about the function. Okay, so one of the more popular structural experimental structure is X-ray crystallography. Okay, so in X-ray crystallography, what you do, you take your molecule and make a crystal. A crystal means like the molecules are arranged in a nice array, in a repetitive array, like one molecule next to another molecule, next to another molecule, and so on. So growing a crystal is an art. I have seen PhD students who spent 5 years of their life could not grow a crystal. That's really unfortunate. It's really an art. Huh? The techniques are being developed, but it's still like really art. So it's a very difficult one. And then once you have the crystal, you can pass a high energy X-ray through it, and you are going to get a diffraction pattern. You're going to get a diffraction pattern. From that diffraction pattern, what you get, like if you are familiar or heard the term called Fourier transform, you get a pattern, what you call in a Fourier space. That means like you get like how repetitive they are, and you can translate it to our normal space. That means like the volume or like phase space where we know the distance and stuff. And then you get an electron density map. So from X-ray crystallography, basically your experimental data is an electron density map. And once you get the electron density map, there you can fit your protein different atoms and you get a structure, atomic model. So that's the X-ray crystallography. Most of the PD deposition is still X-ray crystallography. And the resolution you get is like between 1.5 angstrom to 6, 7, 8 angstrom, depending on like one of the major bottleneck of uncertainty is like how pure is your crystal. If your crystal has impurities and so on, so then the resolution is higher. And then of course, how dynamic is the protein. As we talked before, sometimes the resolution is high because the protein is dynamic. That does not mean that we don't have information there. It's just that protein is dynamic itself. Okay, so next is NMR. So that's another experimental measure. And I think like, I do not know if in Bangladesh you can do X-ray crystallography, but you can definitely do NMR in Bangladesh. I know that NIB has an NMR machine. So in X-ray crystallography, you almost, I shouldn't say do not have any restriction in size, but you can go a very, varied number of how big the protein can be, how small the protein can be. In fact, X-ray crystallography can even do small molecules, But in NMR, there is a size limit. You can only do up to certain big. So the NMR technology, what happens is you take your protein, you replace some of the atoms with a heavy atom. Okay, so let's say you replace your hydrogen with a heavy hydrogen. And then if you replace that, then that atom has a spin. If you put a magnetic field, all will be spin up. If you let the magnetic field down, then they will like slowly recede. And depending on how two atoms are close to each other, how they are coming down will be different. Like in the sense that if there is no two heavy atoms close to each other, no two atoms with spin close to each other, they'll drop down faster. If they are too close to each other, they'll drop down slower. So that gives you a distance restraint, gives you an idea which atoms are close to each other. Okay, so in X-ray, you get a density. In NMR, you get a distant restraint, all the distance restraint. And then you model, and you say, like, I need to satisfy all these distance restraints. What will be my model like? And that's why you come up with an NMR. And that's why in a PDV file, when you look at an NMR, you see like 10, 20 different models, because all of them satisfies those distance constraints. Okay, so NMR, just remember, you are using the distance constraints, and then you are creating a model from the distance constraint, but they have a size limitation. Okay, so the protein modeling actually started with very similar idea. There is another experimental method, which is like the really most popular method, cryo-EM. We are not going there because it's not relevant to our talk today, but I just want to mention that is the most preferred method of solving protein structure, especially if your protein is not too small. Cryo-EM is, is easier to solve structure. You don't have to grow crystal and so on. Okay, now, so just remember in X-ray crystallography, you have the density, you put your protein there, and sometimes what you do is like, what we call like, if I solve the small protein, I know the density. If I solve a big protein, I can take this density and replace that in the big protein. It's called a replacement, molecular replacement. Okay, and in NMR, we have the distance restraints. And as I go through the history of the modeling, you'll see in the modeling, they do follow the same philosophy. So the first part of the modeling is what we call the template-based modeling. In a template-based modeling means you know the structure of a protein, and you say like, my protein is homologous to this protein. So I'm going to transfer some of the structure from the protein that has been solved to my protein, which is a homologous. So that's called template-based or homology, the term that you may have used. So basically what they do is like, if the two proteins are in the sequence level, they are close to each other, then I can transfer their structure. One experiment is probably solved from one to the another. Okay, think about the X-ray. If I know I have the density of a smaller protein, I'm trying to solve a bigger protein. I know the density of the smaller protein and structure in the smaller one. I take that and I put it in the bigger protein. Actually, in most of the cases, it's actually the side chains and stuff that you transfer. So template-based modeling has the similar philosophy. I know something, and I know these two will be related. I transfer from one to the another. And then we have the template-free modeling, which is what you did a little bit, which is called MD simulation. And that means like you do not have any other option. Your protein is not homologous, your protein does not have any homologous, and so on. Until now, MD was the way to do for something or some similar method we call ab initio method. That means like you don't take information from anywhere else. So those are the two approaches, okay? Template-based modeling. This was just outside my office at Purdue when I was doing postdoc. That's how they really used to do, in the sense that, well, this is not, but they're similar structure. The first one was done in 1969. You literally build, this thing was this big. You literally build the structure of a protein by wires and nuts and bolts and so on. Okay, from one, and you literally build. And then this is like, let's say, experimentally solved. And then you start building the protein that you want to solve. I think in this case, bovine alpha lactalbumin was trying to be modeled with from the structure of egg white lysozyme. Okay, so then you start building your wires, and you say like, this part of the sequence seems similar, so I'm going to copy this part with this part. And then somewhere you say like, oh, this part doesn't make sense, so I have to do something different. So basically, you literally build a little bit by little bit, and we still do it. We algorithmically do it a slightly faster way, but that's the first way you used to do it. Okay. So algorithmically, how we do, and until AlphaFold was invented in 2021, the iTaser was the most accurate, but there's still a big difference between accuracy of iTaser and AlphaFold. And the way that iTaser used to do, you take your protein, you break it down into small pieces, and you say like, how does this piece look like? Does this piece has any homolog into the PDB? So you basically don't look for homologs for the whole protein. You look for pieces by pieces, and then you combine them together. So that gave you a benefit that even if you don't have a homolog for the whole part, if you have homologs for the pieces, you can combine them together. So that's iTaser. And if you look at the author list of iTaser, the first author is A. Roy. That's not me, somebody else. Okay. Okay. And then there are lots of other one, but that's like template-based modeling. Okay. Now in template-based modeling, a lot of the time you need what is called multiple sequence alignment. Because what multiple sequence alignment does, it allows you to look for what part of the protein is similar to other protein in a part of the family. And it's actually a very important tool. This is what is the fundamental of AlphaFold. We'll come to that. So the whole underlying assumption, or whole underlying information comes from the multiple sequence, and it does for the template-based modeling too. Okay, so I am not a specialist on multiple sequence alignment. I'm going to skip it. I'm just going to mention, if you have to do multiple sequence alignment, Hammer, though like a BLAST is an iterative search. So BLAST is good for a single searching, but multiple sequence alignment, they may miss something. Whereas Hammer look for, use the Markov chain model. So they are a little more sophisticated, and they'll find you distant relatives. Okay, so if you have, there was like a guideline, like if you have sequence identity less than 50%, you can just close your eye and say like, these two protein looks same. If you want the sequence identity start going below 30%, then you are in the twilight zone. You are not sure if I can transfer from one to another. But again, these are all history now. You don't have to remember, just I'm going to the history. Like when you will be teaching others, you don't have to teach all these. Okay, so template-free modeling, in the sense like when we don't have any information out in, how do we do it? The way to do it was typically MD simulation. You start with some kind of structure, you run MD simulation, and you hope like the structure will form. But again, as I said, the MD simulation can simulate only up to microsecond, maybe millisecond. A protein may not fold correctly within that time. Okay, so that's why, even though ideally you should be able to do it, but because of the limitation, MD simulation is not a good method to predict structure just day and night. People still do it for smaller proteins, certain proteins that you can do it. People has done it, but this is not the way you go, because just of the requirement and the time. Okay. Another very nice software suite is called Rosetta. Rosetta is a kind of a combination between ab initio and between physics-based course and like the knowledge-based. That means like you transfer from other template. It's a combination of both. And it's actually a very powerful, very good tool. It's a huge tool. You can do so many things. There's one way of doing computational biology is to just learn Rosetta, in the sense because they have all the binding energy calculation. They have, their philosophy is slightly different. They start from a coarse grain, then they go to a finer grain, and they go through a finer grain, and so on. And they have their own version of AlphaFold too. It's called Rosetta Fold. So if you want to be in the Rosetta community, you can just live there without doing AlphaFold. You can do Rosetta Fold. And they're coming up with really nice. So that's a huge field. Like every year there is a Rosetta convention just for the Rosetta people, and hundreds of people show up. So you can imagine like hundreds of PIs around the world, they're just doing it with Rosetta. Okay, so that's just, I wanted to mention that Rosetta is another source where you can actually just learn Rosetta and do everything with the Rosetta. It's computationally expensive though, so you have to use the HPC. It's not possible to do it on your laptop, but you can use their server too. But all of this method has their limitation because they're making approximation. Just keep that in mind. Okay, so these were the histories. And when everybody develops their method, they say like ours is the best, right? Like it's always the like a paper, novel method, this, and so on. So that's why the community came up with a competition, which is called Critical Assessment of Structure Prediction. Okay, it happens every 2 years. I participated in for 2 years, well, 2 times, like 4 years. What they do is like, they will go to the experimentalist and they will say, do you have a novel protein? That means a protein that you have solved the structure. That means a protein, there is no homolog to it. Okay, so if you do have the structure, please hold on. Don't release it yet. Okay, so they will just get those structures that are novel, that do not have any homologs, and they'll hold the experimental results, and then they will release those sequence only and ask the community, okay, now you predict, and you'll be given 3 months of time. So after the 3 months, you predicted, and we have the experimental structure, and we will see how good you are. Okay, so it was and it's made the standard for how to critically assess a field. Because we all can say we are doing well, but we have to show up here, and we are going to. And then what happened is we learned that we are not doing well, but we are making incremental improvement every time. Like, okay, this year we know, okay, these were the mistakes. Okay, let's try again. And I took part in when I was at Purdue with one of the group that I was at a postdoc at. We took part in the protein refinement. We actually became second in that category in 2014 or something. But it's a lot of work, because those 3 months you just don't do anything. You just do that, and that's why I don't like to partake in those, because you don't do anything, just 3 months. But this is like really a critically, assessment of structure prediction. So that gives us an idea where we are. Okay, and this was the result, and I'll go through the result a little bit. It's a wonderful plot. This is the result in 2020 or 2020. I think it's 2020 maybe, or 2021. Okay, so what you are looking at on the x-axis is how are the targets. An easy target means they may have homologs, and a difficult target means they don't have any other information. Okay, so from left, easy targets, right, homolog. And then GDT means like whatever you predicted, how many of the residues, if I overlap with the experimental residue, and I can calculate like how many of the residue fell onto each other and how many of the residues did not fall into each other. So I can calculate those distance, and then I can see like what percentage fell into within 1 angstrom of my experimental values. If 100% fell into it, that means like my is perfect, right? And then, so there are like 3 shells. If 8 angstrom, that's a huge difference. So you want your you know, around 2 angstrom, less than 2 angstrom is good. So, and then if you look at different cusp, I think this is like cusp 2, 4, 6, and so on, okay? And you can see everybody does a good job when it's an easy one, right? Because it's a homolog, you can just transfer. And if you look at the initial cusps, you see as it difficults, we were getting around 20% of the residues correctly. And with the, you know, every year, so there's cusp 14, that means like 28 years or 30 years. With the 30 years, you know, we moved up here in the sense that we were able to get around 70% of the difficult residues within an angstrom or 2. Okay, so that's the 30 years of community effort. Like every year you move a little bit and so on. And then came AlphaFold. That's the line that you are seeing. Okay, so no matter where, you are more than 90% time accurate, right? And that's when we say like, okay, we're going to leave everything. We're going to just talk about AlphaFold from now on. You know, there is a valid reason, and that's why we do. And then, you know, we are learning more, like how many things it can do. Okay. And then also, yeah, I forgot what I was going to say. Okay, so that was when this was actually came in 2020, and the organizer arranged a meeting, and they said like, man, either these people cheated or they have solved the problem, because you cannot get that right, okay? And then finally the double check, no, these people didn't cheat, they got that right. And then AlphaFold, some people are calling it AlphaFold 2. That's because there is a version 2 years earlier they tried, which is AlphaFold, and they were not that great. I mean, during the next time they adopted some methods that was actually started in Columbia University, and they adopted it, and that allows that to be. And so now they call it AlphaFold again, but you know it's actually AlphaFold 2. And also, unfortunately, I forgot the name of the professor at Columbia. He's not mentioned, but he's the original idea who said like how to do it. Okay. Okay. So what does AlphaFold? What is the basis of AlphaFold? Like what? Like in template-based modeling, our basis was if I have a known structure, I can transfer that to a known part of the sequence. In template-free modeling, my basis was I can calculate the fundamental physics forces and I can model it. What is the basis of AlphaFold? And this idea came, maybe in late 2010s, like something, I forgot the date. It's called direct coupling analysis. So what is a direct coupling analysis? If you do your multiple sequence alignment, okay, then, you know, you will see certain residues are mutating with same frequency. So in a multiple sequence alignment, what you have the evolution history, right? The older organisms, they have the older version of the protein. The newer organisms, they have the newer version. So you kind of have a history of as the protein is going through the evolution. Then you can calculate like this particular residue, how often it mutates and how is it mutating. And then you can see like, okay, this one is mutating with this pattern. Now, if the two pattern of two residues matches, so that means they must be somehow coupled. Otherwise, why they are mutating at the same rate or same frequency? And the hypothesis is, so if they are coupled, they must be close to each other. And that's why the functionally, if one changes, the other has to change. And that's called the direct coupling analysis. This one came before and people used it to build the bigger protein structures. But this was the idea which first took, like, okay, if I have the distance constraints, maybe using that NMR philosophy, I can make it to a structure. Remember in NMR structure, you get the distance constraints. So in a direct coupling analysis, you still get the direct from the MSA, you get the distance constraints. Okay. Yeah, it's basically simply like if I have these X1 and X3 and X2, all of them are mutated in a correlated way, I can calculate partial correlation. That means like if I take away the effect of X2, what is the correlation between X and X3? So in this case, it's 0. That means these two are not correlated at all. Only these two is correlated and these two is correlated. So that way, you know, X1 and X2 are close in distance. So is X2, X3, but not X1 and X3. So this is like the basis of the one. And so now we have all the P's, right? We run an MSA, and that's why the first step of AlphaFold is running a multiple sequence alignment. That's why you do it. You run all the MSA, you get the distance constraints, and then somehow we need to do the prediction. And that's where the machine learning part comes in the sense that given a distance constraints, how can we translate that into a geometry? In the sense like, I have all these distance constraints, how can you put them together to make a structure? And it was, that was the final block that AlphaFold did and brought all of them together. And, you know, it's really, really accurate. Like it has made our life so easy. So when you get AlphaFold, the another beauty of AlphaFold is it can tell you which part it is confident about and which part is not confident about. So there will be two kind of error. One is called PLDTT, that means like position-based error. So for each amino acid residue, you get a PLDTT score that goes between 0 to 100. And this score is like very similar to the GDT score I said, like how many residue I expect to be within 0.5 angstrom of the original answer, how many residue I expect to be within 1 angstrom, and so on. Okay, so higher the value, that means I expect most of the residues to be within 1 angstrom and so on. Okay, so when you are looking at an AlphaFold structure, that's what you look for. You will have a PLDT score for each residue, and that gives you the confidence, and we'll look at the result when you download it. That gives you the confidence, how well do I know my structure? And that was not before there in any of the previous model. Okay, so yeah, regions greater than 90, you can trust them really. 70 to 90 is, at least your backbone is right. And if it goes below 70, over 50, then you have to be like careful. You have to use your own other knowledge. If it is like less than 50, there are maybe two options. One is AlphaFold cannot predict it, or this is fundamentally a intrinsically disordered protein. That means it does not have a structure. Not necessarily every part of the protein has to have a structure. Okay, certain part of the protein can have a structure, other part not. So it can be either. Okay. What's that? Yeah. Yeah, so certain proteins, what we call intrinsically disordered protein, it stays in a it stays in a flexible part, and they're functionally very important too. So because they are disordered, you cannot predict the structure. So when AlphaFold does not gives a low confidence, it can mean either. It can mean it can be an intrinsically disordered protein, or it can be that we don't know it. Yeah.
What is the specialty of loop generating this?
So loop is defined as like a portion, flexible portion between two secondary structure, right? Now, if the loop is small enough, almost any of the model, and small means like 4 to 6 to 8 residues, any of the modeling thing can do well. If the loop is big, long, 10, 12, 30 residues, at that time, the loop can be an intrinsically disordered part, or it can be slow flexible that it may not be able to model. You may not be able to model. So you have to ask the question first, what am I going to do with the model of the loop, right? I mean, like I give you the model of the loop, let's say, and what are you going to do with it? Let's see if I ask you, what are you going to do with the model of the loop? Why do you need the model part? And then, okay, so let's say for MD simulation. For MD simulation, it's okay to start with any decent start, because MD itself will make it dynamics, and hopefully if you run long enough, it will find the right conformation and so on. So it's okay to start with a decent loop structure. You don't have to have the perfect. Sometimes there is nothing called a perfect structure for a loop or flexible part, because they are so flexible that you don't know what will be the real structure. For MD simulation, you are fine. You start with a decent structure. For the loop, you will be fine. Yeah. Okay, so then there is another error called predicted align error. What is the predicted align error? So PLDTT means it's per residue. And again, these things might feel abstract, but once you look at the result, you'll find it. PLDTT tells how accurate are each residue. And if you have more than one domain in a protein, how accurately they are put relative to each other. That is called predicted aligned error. Okay, so how accurately the different domains are put with respect to each other. I think that's it. So just conclusion, it's outstanding quality. Look at its database. You probably can download most of the proteins there. Sometimes even you do the individual domains, but if you have multiple domains, the orientation relative to the multiple domains may not, AlphaFold may not give you a correct answer, but you will see that in the PAE error, like predicted align error, which tells you how the different domains are. You will see that. And when I gave this lecture to the Thailand, this line was protein-protein interaction. I do not know AlphaFold, how does it do? But there's a paper just came out like last month, or like something like a few months back. They are getting 72% accurate, even for protein-protein interaction, the one that we are doing today, right? We put like two protein chain, and we're trying to like, okay, let's try to build it a model of the two protein. Okay, so they're still best in the class. Nobody else docking program like protein-protein docking does come anywhere close to it. Okay, so it's not as accurate as more than 90% for monomer, but it's still very accurate. Okay, even for protein-protein docking. Okay, I think I'm going to stop here. We'll take a break. Any question? Enough? Yes.
So earlier you mentioned that when we are talking about density, if we know the density of smaller protein, then we put the density to the bigger protein that we don't know. So my question is that as this protein and one is smaller, the other is bigger, they might have structural difference, they might have density difference. So why we are putting the density of smaller protein in the larger one?
Yeah, I should not have used the term protein. It's basically it happens on the residues and few residues only. And when you're talking about the only few residues, or just it's called, or like even at the level of atoms, actually, their structures is very similar. So I should not have used that's my mistakes. Thanks. It's called molecular replacement. You transfer a group of atom from known structure to an unknown structure.
The atom might be similar between these two.
Yes, you know the sequence. From the sequence, you know like the atoms we should.
The atoms is similar in that sense that
Yeah.
we both can be like.
Yeah. So those are sequence is very cheap to do. You know, so that's the first thing everybody does. You get the sequence, you know, like which part. Yes. Any other question? Okay, so let's see. 10:49. I think we should have something waiting outside. Waiting. Okay, so 20 minutes break. We'll be back at 11:10. Okay.